and welcome back to your flipped classroom. Today we are going to be talking about genetics and otherwise how do living organisms pass their traits from one generation to the next. Ready to go? Changing the slide. 3, 2, 1. So let's get some things under our belt. Remember that DNA is a molecule that's going to actually store that genetic code for us. Okay, deoxyribonucleic acid is what that stands for. And there's a hierarchy to it. Okay, it is housed inside the nucleus of eukaryotic cells. If it's a prokaryotic cell, the DNA strand is actually just going to be floating about inside the cytoplasm. And when we talk about DNA in that hierarchy, we can look at the entire strand of DNA, we can look at a portion of that strand of DNA and call it a gene, or we can look at the entire thing all packed together as one unit and call that a chromosome. So if you look over here, here's the individual strand of DNA. If we look at a chunk of it, we can talk about a gene that's located on there, or if we look at the entire thing all wound together, we have a chromosome. Changing the slide. 3, 2, 1. So, with heredity and genetics, genetics is the branch of biology that's actually going to study heredity. Heredity is the actual passing of genetic traits from parent to offspring. So, when we talk about genetics, really the first person to actually study heredity was Gregor Mendel. Okay, he's the one that carried out a series of experiments related to how certain characteristics are inherited in pea plants. Okay, so it's Mendel and his peas. And he was a monk. He studied them. He had his own gardens. He looked at the difference between round versus wrinkled seeds, yellow or green seeds. He looked at flower color, shapes of pods, lengths of stems. He was a phenomenal scientist, and really we do consider him to be the father of genetics. Changing the slide. 3, 2, 1. So, with Mendel's experiments, he did discover that an individual actually can have 2 copies of a gene. You get one of these copies from each of your parent. We call these genes, or these copies, alleles. Okay, so these are the different versions of the genes, and he started to represent these based on letters. He also discovered that one allele is going to be dominant and one allele could possibly be recessive, depending upon the combination of alleles that you have. So the dominant simply means that it is going to be the expressed form of the trait, and we call that the capital letter or the capital allele. The recessive is not an expressed form of the trait, it's a lowercase allele. The only time you get that recessive phenotype is when you have both copies of said recessive allele and there's not a single dominant allele present. So in the example of flower color, purple flowers were dominant to white flowers, but white flowers would crop up whenever there were the lowercase p's put together inside an organism. Changing the slide. 3, 2, 1. So, with these alleles, we can talk about an organism's genotype. Okay, this is the actual set of alleles that the individual has. It's the actual physical genes, and we simply represent genotypes based on the letters then. So you can be homozygous, meaning you have identical alleles for a trait. That means you have either 2 dominant alleles or you have 2 recessive alleles. Therefore, you can be homozygous dominant, all capital letters, or homozygous recessive, all lowercase letters. If you have the combination of 2 different alleles, as in a dominant and a recessive together, you are classified as heterozygous at that point. So, that's the genotype. When we talk about the phenotype, phenotype sounds like physical, okay? Pheno, physical, and that is exactly what it is. It's the physical appearance of a trait, how it actually appears. For example, you have blue eyes. There are purple petals on that flower. That plant has a long stem. That would be the phenotype as opposed to the genotype. Changing the slide. 3, 2, 1. So, the laws of heredity. Mendel also worked on this, and he came up with the law of segregation and the law of independent assortment. What this means is that with the law of segregation, to segregate something means to separate it. So this simply means that the 2 alleles for a trait separate when the gametes are formed. You as an individual have 46 chromosomes in every cell of your body except your sex cells, except your gametes. When your gametes are created, those 2 copies of the alleles separate from each other so that you only have 23 available to pass on to your offspring. The law of independent assortment means that the inheritance of one trait does not actually influence the inheritance of another trait, so that they're separate from each other. They can both be segregated, and then they are independent of that segregation from each other. One gene does not always mean that you get another one with it. Changing the slide. 3, 2, 1. So we kind of call this, since Gregor Mendel was the one who discovered this, we call these very predictable patterns Mendelian genetics. We do this through Punnett squares. It's a simple diagram used to predict expected results of a genetic cross. This also allows us to consider all possible combinations of the gametes for that cross. You can do monohybrid crosses when we're only dealing with one set of alleles or one trait, or you can do dihybrid crosses where we predict the outcome of crossing two traits at once. Changing the slide. 3, 2, 1. When we look at monohybrid crosses, so if you're going to cross two heterozygous frog parents using the traits capital G for a green frog color or lowercase g for a brown frog color, that's what their genotypes would look like. Parent 1, okay, versus parent 2. We set it up with the 4 boxes. Based on the law of segregation, parents can only give one allele at a time, so parent 1 could only give this capital G or this lowercase g allele. Same with parent 2. And then we simply bring them down and over as we fill out our boxes. So when we look at the probability that the offspring is heterozygous, okay, that's a 50-50 chance because 2 of our boxes out of the 4 are heterozygous. When we look at what the probability that the offspring is green, we come back up here. Green frog color means that you have to have at least one dominant allele present. So if you count 1, 2, 3 boxes that have a dominant allele present in them, that means a 75% chance that the offspring will be green. Changing the slide. 3, 2, 1. So here's one for you to practice. Cross a heterozygous parent with a homozygous recessive parent using the traits capital H for long hair and lowercase h for short hair. Consider this. How many of the offspring would have long hair? And also, what is the probability that the offspring will be homozygous dominant? Changing the slide. 3, 2, 1. When we look at dihybrid crosses, we're predicting the possibility of an offspring being born with 2 traits from their parents. Okay? This is a fairly complex thing. I'm not going to go over that in this video. That would take too long. But you can stop here and go watch one of the 2 How to Complete a Dihybrid Cross videos available at haughtonScience.com or at your Google Classroom, and you will be able to see those be walked through. Here is an example of one using cats. If you have any other questions, please come to class prepared with them. Changing the slide. 3, 2, 1. So all of that was Mendelian genetics, where you have very obvious dominant versus recessive traits. We can classify another grouping of genetics as non-Mendelian when they don't fall into those very specific rules. First one we're going to talk about is incomplete dominance. All of the non-Mendelian genetics is going to affect the heterozygous allele combination. When in incomplete dominance you have heterozygous alleles, you're going to see a blend of the dominant and the recessive trait, which means you neither see, in this example, a red flower or a white flower. You would get a blend of the 2 and you would have a pink flower. That is incomplete dominance. As opposed to codominance, again, this is the heterozygous combination of alleles. Both alleles are being expressed at the same time, which means you get spots. So, for example, brown cow versus white cow, you end up with a brown and white spotted cow. So codominance gives you spots, incomplete dominance gives you a blend. Changing the slide. 3, 2, 1. Another grouping of non-Mendelian genetics can be called sex-linked traits. Okay, this is going to occur when you have an allele that is actually attached to one of the sex-determining chromosomes. To be female, remember your 2 chromosomes are going to be both X and X. For males, it will be X as well as a Y. The dominant and the recessive allele are usually going to be attached to the X. Sometimes they can be attached to the Y, and they are then written as superscripts, okay, as almost like exponents above the letter. Like I said, most frequently the trait is going to be associated with the X chromosome. So females are the only ones that can possibly be carriers of a gene, because for them, if it's a recessive trait, for them to be affected with it, both of their X chromosomes would have to have the recessive allele. For men to have the affected trait, if it's a recessive allele, or even if it's a dominant allele, since they only have one X chromosome, the moment that chromosome has the affected trait on it, the male will be, their phenotype would be showing whatever that trait is. This tends to be with like color blindness, male pattern baldness, hemophilia as well. Okay, changing the slide. 3, 2, 1. We can also have a category of genetics called multiple alleles. So these are actually genes that could have 3 or more possible alleles that come together to make up a trait. Primary example of this is your blood type. Okay, there's actually 4 types of blood out there because there are 3 different alleles that code for it. So you can be type A blood if you have both A's present, or an A and an O. You can be type B for the same reason. You have both B's, or you have a B and an O. You can be type AB blood when you have one of each. The only way you can be type O blood, though, is when you have both alleles as the O blood. This is kind of the recessive blood trait. Changing the slide. 3, 2, 1. So, meiosis. This is a part of cell division that is responsible for making your sex cells. Okay, this is the division that actually produces the sperm if you're a male, or the egg if you're a female. Through meiosis, you're actually going to produce 4 genetically unique cells that are haploid. Haploid means it contains half of the number of chromosomes than a regular cell would. Like I said earlier, your somatic or your body cells have 46 chromosomes. From these body cells, this is where meiosis would occur to then produce your sex cells, and by reducing it in half, your sex cells then have 23 chromosomes. So the important thing about meiosis that you remember is that this is also where your genetic variation increases. Changing the slide. 3, 2, 1. With meiosis, you also run the risk of chromosomal mutations, and these are mutations that affect a huge portion of your DNA. These are the 5 different types of them. You can have deletion, where a portion of it is just completely lost. You can have a duplication, where you accidentally copy it twice. Non-disjunction is when, as your chromosomes are splitting, you take a portion of one extra part with the other half. Inversion is accidentally flipping the ends of different sections of chromosomes, and so they're inserted in backwards. And then translocation is the chance of chromosome fragments moving from one chromosome to a completely different one. All of these can have very catastrophic effects on the organism as a whole. Changing the slide. 3, 2, 1. Here are some more examples of this. If this is your original chromosome, here's a mutated chromosome, so you can see that that's a mutation. Of duplication, the one below here, you can see it's non-disjunction. And then to the right, this is translocation, where you have a portion of one chromosome getting copied over and switched with a completely different portion of another chromosome. Changing the slide. 3, 2, 1. So we can use different techniques to analyze our chromosomes for this very immediate identification of how many chromosomes do I have and what's important. So when we look at a karyotype, okay, that's going to be this right up here. It's a visual representation of your chromosomes, and it can show you that a genetic disorder is present. If you look, you have 22 pairs, and then you have your sex chromosomes right here. You should have 46 chromosomes total, 2 above each number, and either 2 above the X, or 1 above the X and 1 above the Y if you're a male. The moment you are missing a chromosome, or you have extra copies of them in a karyotype, you would be able to quickly see if you have a genetic issue that needs to be investigated further. A pedigree is a visual representation of how traits are passed from one generation to another, so we can also identify people who are affected with sex-linked traits, autosomally linked traits, carriers, and things like that. Changing the slide. 3, 2, 1. So here's your chance to check your work from that monohybrid before. You should have received something like this, along with, you should have seen that it was 50% of the offspring would have long hair. And then 0% of the offspring would actually be homozygous dominant, because at no point do you actually have both dominant alleles present in one of the offspring. And that is it. You have made it through the end of our genetics material.